So this is part 2 of the digestive system cell types thing. And to preface this, in case you're coming to this video from not part 1, which, what are you doing? We're just going to kind of go over the cell types of the digestive system from the perspective that if we know that the physiological work of our body is done by our cells, then understanding the cells of a system can shed light on the system in its entirety. So it's a bottom-up approach to learning physiology rather than a top-down. And for certain things that can be super useful because they're usually taught top-down, where you get this overview of a system and then you narrow down on little bits of it. But that approach doesn't always work for everybody. So let's do it differently and we'll see how it goes. Okay. So I'm going to try and make this a little bit tidier than I did last time by using some lines. Oh, I only get to have one line at a time all of a sudden? Okay. What is happening? I'm frustrated. Okay. Why don't I draw a line and then I'm going to try to do something else that's not a line and we'll see if this line stays here. No? Ugh. Okay, so it's apparently only vertical lines that are naughty. We can have horizontal lines. This is bizarre. Maybe if I don't make the line go all the way to the edge of the screen, that will do something. Okay. What a fiasco that was. All right, so we're going to do the rest of the regions. And we don't have that many to go. So after the duodenum is the jejunum, and then the ilium, and then, well, we'll do the appendix because why not? Okay, so that's the areas. Let's dig into the cell types. And remember I said there was going to be some redundancy because of course the small intestine is a continuous tube that gradually changes over the course of its length. So there's not really any sudden stops or starts so much as there are like transitions. So similar to the duodenum, in the jejunum, we're going to do enterocytes, which are the columnar epithelial cells that function as the epithelial lining for the entire intestinal mucosa. And that's going to be true literally for the rest of these as well. So let's write enterocytes again. Well, except for the anus, not that. So with regard to the function of enterocytes, let me see if I can make another line without it automatically erasing. Cool. I'll do functions in green. Okay, so our enterocytes, remember they have a brush border, so they're going to make brush border enzymes. And they actually do this pretty much all the way through. They just reduce in number distally because as you approach the end of the small intestine, you don't need the brush border enzymes to the same extent because, like, think about chyme in the duodenum arriving at the jejunum. It's going to be a mixture of breakdown products from all those macromolecules and, you know, the brush border enzymes like the lactases, maltases, dipeptidases, carboxypeptidases, etc. Are going to be pretty useful there because we actually have monomers, or excuse me, dimers to break down into monomers at that point. But by the time chyme gets to the ileum, and specifically the end of the ileum, almost all of the absorption of that kind of thing and digestion has taken place. So we don't really need that many brush border enzymes by that time. So when I was talking about the small intestine and its sort of gradual transitions, the enterocytes in the small intestine do all create brush border enzymes, but just not to the same extent, and they're not as numerous as you move distally in the small intestine. What increase distally are the goblet cells that are alongside the enterocytes. So I'm just going to do brush border enzymes, i.e. carboxypeptidase, etc. And the enterocytes also, of course, function for nutrient transport. Okay, so that's enterocytes' job. And then increasingly, as we move down, they have different functions. And I want to asterisk the colon enterocytes. No enzyme function. And they don't really do anything in the way of nutrient absorption much either. The point of the colon is pretty much to suck every last little bit of water that we possibly can out of the feces so that it's nice and compacted and a solid, easily passable mass, and absorb anything that's coming along with that reabsorbed water, including things like the vitamin K that is produced for us by our friendly little renters in our colon, bacteria. So speaking of that, well, actually, let's do goblet cells first. There are goblet cells in the appendix still. So goblet cells are unicellular mucus glands. Why did I write that? Let's try that again. Gland is spelled like that. There we go. So there aren't very many glands that are unicellular, but this is one. And so they just sit in the columnar epithelium that lines the GI tract and make mucus to help lubricate the passage of whatever it is that's passing. So anything from chyme to feces counts. And the function is going to be the same for the rest of those regions as well. So I'm just going to make ditto marks for the function because it does not change. So I heard myself. To add to this, we do have to add one thing here. And obviously there's going to be mucosa-associated lymphatic tissue all throughout the small intestine, but I do want to emphasize the difference between generalized malt and what's in the ileum, which is mucosa-associated lymphatic tissue in the form of Peyer's patches, which is a little bit different. Let's see how visible this purple is. Maybe I'll do that. Oh yeah, that's fine. So since we know that the function for the enterocytes and goblet cells is the same, I'm just going to erase this. So a Peyer's patch is lymphocytes in a group or clump. I like the word clump. For immune surveillance. So because of the ileum's proximity to the cecum, which is part of the colon and a place with abundant bacterial life, you want to keep an eye on all that stuff, especially in the small intestine. So that's really what the Peyer's patches are for. They are lymphocytes. They're T and B cells. And they're going to be a hodgepodge of memory cells, helper cells, etc. So the appendix is a little bit weird because it also has abundant lymphatic tissue in it, which generally has the same function, of course, as what's in the ileum. Lymphatic tissue anywhere always has the same function, which is just keep an eye on whatever it's positioned in so that we don't have bad guys taking over in the form of pathogens or what have you. So if you look at the cecum, it's a blind pouch. So from the ileum, the ileocecal valve is above the bottom of it. And so feces from the ileum just kind of like drops into the cecum. And at the bottom of the cecum is this little tail, which is the appendix, aka the vermiform appendix. Vermiform just means it looks like a worm. So vermiform literally means worm-like. Also a great insult. So if you want to call somebody a mean thing and you want to sound smart about it, refer to them as vermiform, meaning worm-like, although worms are cool. It might be insulting to worms, so just be careful. So there's a lot of debate about what the appendix actually is, because in modern humans, it doesn't really appear to contribute a whole lot to digestion. Like, it's not a special nutrient pouch, and it doesn't really appear to do anything at all except for collect and crystallize poop in it, which are real things. They're called fecal concretions. They are basically old feces that got in there and couldn't get out and has had all the water sucked out of it and is basically crystallized. And yes, if you look at an appendix histologically, you can sometimes see these poop crystals, fecal concretions in the lumen of the appendix. So they exist. I've seen them. So because of the hyperabundance of lymphatic tissue and the fact that feces and therefore the bacteria that live in the colon in and amongst feces can get in there too, regardless of what it used to be, it appears now to be a vestigial pouch. So vestigial meaning a leftover evolutionary feature from something that we used to have but don't anymore. And it may also play a role in immune surveillance because it's largely lymphatic tissue and not a whole lot else. So maybe it's like a little police station for your colon where there's extra lots of lymphocytes there, keeping an eye out for things, but no one's really sure. What we do know is that it's not mission critical for digestion. So if yours gets angry and needs to be surgically removed, like mine did, aside from healing from surgery, there is no alteration of function whatsoever. Okay. Colon also has lots of lymphatic tissue, both in the mucosa and in the submucosa. So let's just same function. And then same function for the goblet cells as well. So there's really nothing special about the colon other than there's a lot of goblet cells. And that's because if you're going to suck all the water out of feces, it's going to get harder and harder. And that's good, except for you need mucus to lubricate the passage of it out of you. So what you don't want is fecal impaction where like basically the poop gets stuck and too much water has been taken out of it and then stuff builds up behind it. That is a thing that can happen with certain disorders of gut motility and other things. But usually it doesn't because we've got goblet cells and nice gut motility and mucus lubricating the way, and then everything goes fine. So that's the deal with the colon. There's something else I was going to say about that, but now I've forgotten what it was. Oh, well. Okay, and then anus. So this is different because similar to the esophagus, it has no function other than transit. So if the esophagus is going to function to transport chewed up food from your mouth into your stomach, the anus is going to be functioning only to transport feces out of you and me. So for that reason, it has stratified squamous epithelium instead of columnar epithelium, and that's going to basically perform the same function as it does in the esophagus, which is that sometimes the things that are abrasive pass through the anus. And so if you have a nice thick layer of stratified squamous epithelium, if you lose a few cells, it's not a big deal. Stratified squamous epithelium is highly regenerative, and the top cells are supposed to slough off and die anyway. So not a big deal if we lose some due to the passage of some especially abrasive poop. And then that's really about it. So obviously I'm leaving some stuff out here, like all of them have smooth muscle making up their muscularis mucosae and their muscularis externa. All of them have a submucosa, which is dense irregular connective tissue with nerves, lymphatics, arteries, and veins in it. But you know the general function of all of those things. So for me to go and do a deep dive into the cell types that make up veins, for example, is not really a good use of our digestive system time, because veins aren't specific to the digestive system, and neither are fibroblasts for that matter, nor are smooth muscle cells. So those are things that you should know are there, and you should know their function. So smooth muscle cells contract to accomplish peristalsis, segmentation, and churning. But beyond that, it's not worth spending the extra time to line up their functions here. So that is for the cell types of the tubes. We do want to talk about the function of some of the accessory organs. So let's do that. But again, some of these are going to be redundant, and in other cases, we're going to kind of gloss over some stuff because it's just beyond the scope of the class. For example, if I wanted to describe all of the things the liver does, we'd be here all week, and we don't have time for it. So let me, oh no, Tortoise is doing his thing again. I'm going to take a screenshot of this and then pivot over to accessory organs, and then we'll be done. Okie dokie. So liver first. No, pancreas first. So for pancreas stuff, obviously, we're going to have a lot of different cell types. I'm going to stress the digestive ones here over the endocrine ones, but of course, you know that it does both, right? And I'm going to fetch the tortoise again because he is trying to dig a hole in wood, and it's so loud. Sir, what are you doing? Come with me. Okay, now I just have to not forget he's in the couch. Okay, so for pancreas, we have two overarching cell types, and those are going to be endocrine and exocrine cells, right? So we have the and because we don't actually emphasize telling the difference between alpha and beta cells in our class, because although you can histologically stain and differentiate them, Histology guide doesn't really have much in the way of that, and it's not the most important part for understanding pancreas function, so I'm kind of going to gloss over it a little bit, but that's the general idea. So alpha cells produce glucagon, beta cells produce insulin. So quick aside about the beta cells and the insulin thing, certain kinds of diabetes appear to be autoimmune in that the immune system comes to regard the beta cells of the pancreatic islets as a pathogen for some reason and attacks them and destroys them. So the reason that those folks can't make insulin is because their immune system is waging a war on the cells that produce it for some reason. And that's becoming better and better understood as time goes by. So I think within our lifetime, we'll have some sort of solution for that, which gets rid of type 1 diabetes. But at least for right now, we haven't quite gotten there. So people still rely on insulin. And because of the United States healthcare system, people in the United States die from not being able to get insulin, like on the regular. Most notably, not long ago, I think it was a couple of years, a person made a Kickstarter or a GoFundMe or something to try and raise money to pay for their insulin because they couldn't pay for it anywhere else. And the fundraiser did not get fully funded. And yes, the person died because they couldn't buy insulin. So, not to be a bummer, but we have a very bad broken healthcare system if people are making GoFundMes to get insulin and then dying as a result. Like, that's not chill at all. So
I have a question real quick.
Go ahead.
So I was listening to a podcast recently, and it was detailing the different types of insulin and how one is used by our healthcare system or required, and then the other in, I think, Europe is a cheaper version that works just the same.
Yeah, I've heard of this too. Is it the difference where there's like a long acting and a short acting one? That's the one I've heard about.
I'm not too sure. It's been a while.
Okay. I'm also not sure, so that's fine. Anyway, what was your question?
Just wondering if you knew anything about it.
Oh, I don't know much other than what you said. And there's like certain rules about drugs that are under the auspices of protecting the United States citizen from receiving poor quality drugs and suffering injury as a result. But given the success that people have in ordering drugs from overseas and treating themselves, which is a thing, it seems like those laws are probably more about protecting the financial interests of American pharmaceutical companies, unfortunately. Like, I'm not one of those like conspiracy theory, like, ooh, big pharma is going to kill you type of people. But I also think we have a broken healthcare system that prioritizes the needs of billionaires. So,
What I additionally to that point, what I did hear was that Europe has a certain section of government that's purposely meant to investigate the cost effectiveness of certain drugs. Somewhere, I don't know if it's Britain, I don't know if it's certain areas, but I know the US doesn't have it.
mm-hmm.
And is probably one of the factors that are are causing the lack of the inability to purchase needed insulin or any other drugs of that nature.
Yeah, absolutely. I mean, I would agree. I would bet that multiple nations have something along the lines of that, or in the context of the EU, probably the EU is like, okay, well, you have this and you have this, so let's just make a big one. But yeah, I would totally agree with you. And there's other things that like are evidence of that as well that aren't insulin related. Like obviously the insulin example that you and I just discussed is a big one. But that's also the reason why a couple years ago there was a big scandal where a dude named Martin Shkreli purchased a pharmaceutical company and jacked up the price of an antiparasitic drug that is also used in supportive care for people with HIV and AIDS. And he raised the price of it 4,000%, creating a situation where it was literally life or death, like the insulin sitch. He is in prison currently, but not for that. He's in prison for defrauding investors. So he's in prison for bilking rich people, not for endangering the lives of people by depriving them of medication. So everything's bad, guys. Not good. I'm hoping that you will matriculate from A&P with me and become ethical clinicians, all of you, regardless of where you end up, and you will help fix that to whatever extent you're able. That's my goal. Because I think that everybody deserves to have healthcare, and it sucks when that's not true. But yeah, so thank you for the input on that. I didn't think about that European thing, but yes, now that you mention it, I do recall the same thing. I'll go do some digging and look at more of it because I'm curious about that now. I also looked up insulin the other day for an unrelated reason, for just work stuff, and now Instagram won't stop advertising dog insulin to me. It's like every other ad. It's almost to the point of where like I feel like it's threatening, where they're like, whoa, it'd be a shame if your dog got diabetes, wouldn't it? All right, so in addition to alpha cells and beta cells producing glucagon and insulin, we also have cells in there that produce stuff like insulin-like growth factor, but that's less relevant to the digestive system function. So let's pivot to the exocrine function. So we have two other populations of cells, not just one. So these are the cells that are arranged into acini, as per the first practical. And then we also have ductal cells. Which, as it sounds like, they make ducts. But in this case, they're not just for being a duct, they're also for a specific purpose. Okay, so as we mentioned previously, with regard to the exocrine function of pancreas, it does a lot of stuff. So we have, well, I'm not going to say pancreatic because obviously it is, and it's just a waste of a word. Lipase, but from the pancreas. Alpha amylase, but from the pancreas. Trypsin. Chymotrypsin, etc. And these are, remember, these are precursors. They're not active until they're secreted into the duodenal lumen, because we don't want them to become active inside of our pancreas and start digesting our pancreas and turning it into goop inside of us, which it will if these are activated. So instead, once the pancreatic juice gets squirted out of the either major or minor duodenal papilla, enterokinase, which is secreted by the duodenal cells, is going to activate trypsin and chymotrypsin. So that's kind of the deal. And then ductal cells, in addition to being a duct, which is just a tube that carries exocrine stuff, they also secrete bicarbonate and phosphate rich serous fluid. And that mixes with the above exocrine products to form a healthy blend of digestive juice that we collectively call pancreatic juice, which, you know, it's a nice brew of all these things. And then if that is required to be secreted along with bile because lipids are detected in the duodenal lumen, then you would see a mixture of that stuff and bile coming out of the major duodenal papilla. But if we only needed pancreatic juice and we didn't need bile, then we would use the accessory pancreatic duct, which links up with the minor duodenal papilla. So it really just depends on whether you need to include bile or not include it. So the pancreas is magic. It's very special and fancy. And it does a lot of different things. Okay, liver. So I like to make a dad joke about this, which is that they call it your liver because you need it to live, but it's true. So there you go. Obviously, hepatocytes have a lot of functions. So just now when we were looking at the urea cycle to try and talk about where the urea that we pee out comes from and why we want to get rid of it and why we keep some of it, from Darren's excellent question. We talked about the urea cycle, where ammonia is combined with carbon dioxide and then sent through some cyclical reactions where we spit out fumarate and urea at the end. That happens in liver cells. So that's one of the many things they do. So let's kind of make a list that's a summary. Obviously, they use hemoglobin breakdown products to create bile. So let's do bile synthesis. And I'm emphasizing that because the gallbladder stores bile and concentrates it, but the gallbladder does not make the bile. So this explains the fact that we can live without a gallbladder, but we can't live without a liver. Cool fact about the liver, by the way, it is derived from epithelium, ultimately, which means it is regenerative. So if I were to say donate a hunk of my liver to somebody else, which is conceivable, I over time can get back some of the tissue that I lost because my liver will just grow back to some extent. So it has this little bit of this superpower that other organs don't really have. Okay, so biosynthesis is one, and then we have the hepatocytes doing all kind of other stuff. So urea cycle, it's going to turn ammonia into urea. ETOH is ethanol metabolism. So when you imbibe alcohol, which is ethanol, it needs to be processed to make it less toxic. And so your liver does that. And interestingly, one of the ways that your liver does that is because the reason that alcohol is toxic is because it creates free radicals, which are little compounds that have an unpaired electron, and they really hate unpairedness. Electrons like to be in pairs. They like to be, one of them has an up spin and one of them has a down spin. Don't ask me what that means. I am not a physicist. I imagine it's about twirling somehow. But it turns out when they're not paired, they're angry about it. And so they will go scavenge friends from whatever's around. And in living systems, the result of that is that they destroy the molecules that make us and do things, and it's bad. So that includes DNA, for example. So free radicals are also dangerous for creating dangerous mutations. So we want to get rid of those. One way to get rid of them is to make and use things with natural antioxidant function and or manipulate other compounds to give the potentially electron scavenging compound something to take an electron from for free, and other stuff as well. So another cool thing that hepatocytes do is they have lots of peroxisomes, which are like a lysosome, but they are full of hydrogen peroxide. There's a fun experiment that I used to do in my job at PCC that involves taking some like chicken liver, I think, or beef liver, and you do some stuff to it to give the hydrogen peroxide something to react with, and it actually bubbles and fizzes as the hydrogen peroxide is produced and used, which is neat to see. So there's that. Let's see, what else does it do? It detoxifies various things, including ethanol and drugs. Vitamin and mineral storage, and in some case synthesis as well. So it can transmogrify things into other things or decide to store stuff. So for example, the liver like traffics vitamin K that it gets from the colon into using it to make clotting factors. It's one of the things it does. It also rips the iron off of heme groups and is like, well, we don't want to poop this out because it's like useful and stuff. So we're just going to save the iron, and then we can give the iron to the bone marrow when it needs to make new red blood cells. So it also makes a carrier for iron called transferritin, as well as many other proteins that you would find in the plasma. Oops, I can't spell albumin all of a sudden. I'm going to run out of room for that word. Let's try this again. Angiotensinogen. Lots of things ending in ogen, as a matter of fact. The liver is the prime source of ogens the body over. So, oodles of functions, that's just a few of them. The ones that I picked to share with you are because they're the ones that have like some direct relation or other to things that we've discussed about in class. But hopefully what's clear by now is that due to the many, many, many functions of the magical organ that is the liver, we get to stay alive, and that should drive home the importance of being nice to your liver because you need it to live. Okay, and that is that. So I'm going to take a screenshot of this too. It should be easier because it doesn't take up the whole screen. Oh, and by the way, just really quickly, since I didn't mention it, all of these functions are performed by hepatocytes, which is just the major dominant cell type of the liver. There are a couple other cell types in the liver that are like things like specialized macrophages called Kupffer cells and whatnot, but those aren't things for you to learn for histology, so I'm not going to belabor them here. But in general, hepatocytes, which are the majority of the cells that you see when you look at the liver histologically, are doing all these fancy, amazing things. So that's that.